for today, so take it away.
Okay, so this paper is Female Choice and Pre-Existing Bias, Visual Cues During Courtship in Two Schizocosa Wolf Spiders. So first, what was their primary question? What were McClintock and Hewitts aiming to do?
Like, is there selection on, like, these secondary, like, sexual characteristics, and then, like, is there, like, a pre-existing bias? Some two things.
So they start off talking about female receptivity and how a female responds to male signals. So what were some of the things they said impacts if a female responds to the male signal?
Are you referring to like the 1, 2, and 3 and 4?
Yeah, I
things?
usually say that there are 4 things.
I mean, I can just read them if it would be.
Yeah.
But I mean, whether the signal was successfully generated, whether the signal was successful at reaching the female through the environment, and whether the signal was received by the female once it reached her, and the decision of the female to act on the perceived signal.
And then McClintock and Hewitts used these 2 spiders as their, like, why were they using spiders? They said spiders are a good model system, but why is that? Like, why were they using these 2 spiders? Why were they interested in?
As a whole, I think they said spiders were a good model system because the females are potential predators and mates, and then they specifically used this genus of spiders, specifically the 2 species, because they're geographically sympatric, and there were some other things too why they picked these specific species, and they're geographically sympatric but occupy different microhabitats.
And they also use these 2 because Acreata males have the leg tufts and Rovneri don't.
Yeah.
So that's what they were looking at specifically was the interest in leg tufts and the pre-existing bias. So they needed one that didn't and one that did.
And what else is interesting about these 2 species? Yeah, they're virtually identical, and what separates them?
And the leg.
Not just the tufts, but also the behavior.
Yeah.
So these are what we commonly refer to as cryptic species, and what it means to be a cryptic species is that you look the same, but you stay reproductively isolated by some other means. In this case, the other means is the behavioral means. Sorry, were you about to
I had it on my list, yeah,
Okay,
but no, that's fine.
Yeah. So there were a lot of good reasons to use this particular genus in this way. So many reasons.
So we kind of talked about how McClintock and Hewitts were interested in the leg tufts, and so that's what they were manipulating on the spiders, right? They were using videos to manipulate the leg tufts, the presence of tufts, to investigate the pre-existing bias hypothesis. So they were kind of predicting that female preference precedes male traits, and that male traits evolve as the means of exploiting these pre-existing female preferences. That's what the pre-existing bias hypothesis is. So they were saying that tufts evolved after female bias. So what did they use for their video stimuli to do this? Like, what were their experimental videos?
Control Acreata, Acreata with no tufts, Acreata with enlarged tufts,
Rovner, it's named after a guy named Jerome Rovner.
Control, which means that they had no tufts, basically, and then one with additives.
And then they used a blank screen just to make sure the screen itself wasn't like messing with the females.
Did you guys go and look at any videos of these guys doing their thing?
I don't like spiders, so no. Absolutely not.
Oh, well, now I definitely have to do this.
Show it. I'm actually
You had
curious.
that other, like, article up or whatever, and it had, like, pictures of, like, up close to spiders. I was like, ugh. Let me just scroll through this.
So this is Schizocosa ocreata.
Writing's not that easy.
I'll make it really big.
Grammarly can help.
Way bigger, yeah.
This sentence
clarity.
undermines the
Where's my
writer's
cursor?
message, and the word choice is bland.
Just go straight down.
Oh man, I can't see.
Cutting-edge technology helps you
Skip
craft
ad,
You're
skip
there.
ad.
There it is.
Ah. So this is Ocreata, and you can see the big tuft on the front leg. These things live in leaf litter, and so it's hard to see them sometimes, and they can't see very far in the leaf litter. And so they have to make themselves obvious, and they communicate through these vibratory cues that then the leaves themselves serve as the transmitter of these vibratory cues, and they're picked up by
use
humans.
those vibratory cues?
Hmm?
Do both species
The
use
paper
Yes.
said
Oh,
they
they
do, but
But it-
Ocreata developed the waving arms because their vibratory signals weren't as
Yeah.
picked up because of
Because of
their
their leaf litter.
microhabitat.
Yeah. So leaf litter is a really difficult environment in which to transmit a signal. That goes back. That's why they talk about those things in the introduction, what the tasks of a signal are.
Why do the females eat spiders? I guess it might be a dumb question, but like,
Spider has everything spider needs.
Aren't females like bigger?
are they bigger?
In this case, there's a slight sexual dimorphism in size, but it's not huge. The main sexual dimorphism is in the color pattern and presence of tufts.
That's interesting.
Hold on.
Oh, got another one.
Of course, you got another one. Let's go back. Let's see if we can find Ravnarai. These videos clearly were made before YouTube was a thing.
I don't know how you'd catch those things. They'd like haunt my nightmares.
That I hate that.
What?
The close-ups
Its face.
with fangs.
Yeah.
Fangs and
It's got some big fangs.
the big eyes. So when they talk about receptivity, this is what they're talking about. This is a male.
Nice.
That's weird. This
No,
is
you
a
have to
male.
play it 'cause he's turning around.
This is a male and female.
That's
music,
the
that was a
female. That's
great
what they were
music
talking
choice.
about with the
The
female
live
res-
tests.
Yeah.
Yeah.
Yeah, it's two clear
Mm,
boxes,
circle.
and the male's here, the female's there, and they're basically videoing her behavior to then go back afterwards and
Yeah.
score it according to those receptivity
Mm.
things. I'm gonna just play one more for you. So you've heard now a couple of the vibratory displays. Stridulans is unique. It's another genus, another member of this genus, and it's unique in that it has a file, kind of like the file that's on those moths that we talked about that jam the bat signals, and so their vibratory signals are substantially different than the ones that you've seen so far. That jank, 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 that's a projection going across a file.
I did not like that noise.
Hmm?
I did not like that noise.
It's interesting.
But you can see why this vibratory signal is different than the Ocreata vibratory signal, and so females would be able to pick up on that.
Oh, it's interesting.
Avery's about to
So
crawl
what
out
are
of his
those?
skin.
Their, as far as their anatomy,
Hmm?
is it their anatomy?
These
is
are
it just the
pedipalps.
pedipalps?
That's the male copulatory organ.
Okay.
The male copulatory
And
organ.
they're just scraping it along the ground?
Yes. So the male copulatory
Yes.
organ on the males is on these things called pedipalps, and they actually weave a little web and then they deposit sperm on it, and then they turn around and they like dab the tips of their pedipalps in, and it sucks them up into the pedipalps. It's called loading the pedipalps. And then the copulatory structure on the female is on the underside of her abdomen. So when the male goes to mount the female, he'll mount her from the back, but then he'll lean over and get under her abdomen to basically get access to her copulatory organ, her epigena. And basically it fits kind of like a lock and key, and he basically provides sperm. He'll do it from one pedipalp on one side, and then he'll shift to the other side and do it on the other side, and just keep flipping back and forth until either she's had enough of him, or he's basically transmitted all the sperm that he has loaded in his pedipalps, has been transferred to
So
her.
where does the sperm come from then?
He produces it, so
But like,
so on the male and the female, if you're thinking of spiders as being shaped kind of like this, there's a genital opening on both the males and the females. In the female, the genital opening is very elaborate in many cases. And so let's go.
it's okay. I was just curious.
Yeah,
I
no.
don't really want to see.
I kind of see.
So this is the underside of the abdomen,
We cannot
and there's
see-
a
we
little.
can't see it.
Oh, you can't see it.
It's okay though.
No, it's fine. It's fine.
I mean, we can see it. You can just put it back on your screen.
So there's this little slit right here. This is the genital opening of the male, and the females have a very different genital opening, and it has a structure to it. And the structure is diagnostic. It's very important for the anatomy. This is the male pedipalp, and you see it's got complex structures to it as well. And there's a little tube. Let's see if they show it in any of these figures. Maybe they don't. Yeah, oftentimes. So this thing actually expands. They push hemolymph into the pedipalp, and this pedipalp kind of unfurls. And then there's a little projection that's like a little tube, like a little needle, and that's where the sperm come out. So they suck the sperm up into that structure, and then they eject the sperm out of that structure. But in order for them to mate, the male's pedipalp has to fit inside the female's epigenum.
Why
And-
doesn't
Hmm?
why isn't the sperm just produced
Yeah.
in the pedipalp?
Because your gonads are in your abdomen. And so the male produces the sperm in the abdomen. The opening is then therefore on the bottom of the abdomen, but they, for whatever reason, evolved an intromittent organ
Yeah.
that is independent of that is on the tips of the pedipalps. And the males have these really elaborate pedipalps, whereas females have really simple pedipalps. It's just like another little mini leg. And one of the reasons that we think that this might have evolved this way was actually as a species isolation mechanism. In order for the male to mate with the female, he has to have a structure of the pedipalp that fits with the structure of the female's epigenum.
Is this common in all spiders? Like this?
Yes, this is the way all spiders do it, for the most part. And the reason that these are called
Oh, so that's why it says the Ocreata and Ravneri are reproductively isolated by their courtship behavior. So that's just talking about their.
that's about
That's
oh,
the
that's about
behavior.
okay. That's about the behavior.
because when
okay.
you look at the female epigenum, you will see that the female epigenum of them look exactly alike. And so, and the reason that they're called schizocosa is that in some species, this bottom portion, the transverse piece of the epigenum, is split, and that's what schiza means. Like a schism, it's a splitting, a dividing of something. And so what you'll see is that these species are really hard to tell apart because their reproductive organs are very similar to one another. So that really the only thing separating them is
Tufts.
reproductive behavior, and in some cases tufts on the legs. And if you knock out, I told you about this in my office the other day, if you knock out a female with CO2, basically deny her of enough oxygen that she passes out, and then take the CO2 off, and then put a male in there of another species, he will mount her and start mating with her. And when she comes to, she'll notice, oh, there's a male mating with me. I must have consented to this at some point, which of course you hadn't. But she'll continue letting him letting him court with her, mate with her, and what they will produce is they'll produce offspring that are hybrid that survive just fine. They grow to maturity. Now what happens is their courtship behavior is all messed up because it's a mixture of one species courtship behavior and another species courtship behavior, such that if you put them with the parental, either of the parental species, chances are they're going to be eaten because the parental species won't recognize the courtship behavior of the offspring. But that's how compatible they are. You can hybridize across species as long as you can get the female to put up with the male long enough to get sperm into her. So, yeah, that's one of the reasons that this genus is interesting to work with. And then this is just a variety of the different variations. This is from the Stratton paper that I put up. The variations in the tuftedness of the different species. So anyway, that is that, and I'll do Avery the favor of removing the image from the screen. All right, continue, Elizabeth.
Okay, so the female spiders were presented with the video stimuli, and evidence suggests that the females respond to the video males the same way that they respond to live males with the same frequency. But the females were shown each video in a randomized order. So why did they randomize the videos?
Wasn't just like make sure that they weren't like.
Picking up on any patterns,
Mhm.
potentially.
Yeah, so they randomized the videos to control for habituation or priming effect, so that way the females didn't like get used to the videos and stop responding to them.
Yeah, so you might get used to them and stop responding to them because you learn after a while, oh, well, that's not a male. Or it might just, oh, I've been exposed to all of these videos, and so you get hypersensitive to the video, and they wanted to test for that.
So then how are they measuring female response?
It was off of like a kind of a point system, for lack of a better word. Just kind of 0 to 3. And
That was for,
Well, they did both. They did absence and like if they were receptive or not, and then they scored it.
yeah.
And then also latency, but that was.
Yeah, I was gonna say there were 3 variables for how they measured response, and that was presence or absence of receptivity, and then a scored response based on recognition, aggression, and receptivity behaviors, and then latency, which is the time between the first orientation to the video stimulus and the time they observe the first receptivity display. So then what are some, we kind of saw it in one of the videos, but what are some examples of how female receptivity is viewed?
The
slow turn.
Settle the lower abdomen and cephalothorax to the substratum, lower cephalothorax, raise abdomen and leg extension.
So then table 1 shows how they scored those behaviors to show like when they were most receptive, and it says that after one of the level 3 behaviors is usually when mating occurs.
I like the term slow turn.
Yeah.
Seems like something that would happen with Barry White on the stereo or something.
So then they did a phylogenetic test, and why were they doing this, like making the phylogeny?
Aren't they just trying to get a map of the tufts in a way? Like they created it says here the presence or absence of tufts was mapped onto the resulting cladogram to determine the polarity for that character.
Basically, weren't they making the phylogeny so that way, I mean, they were testing for pre-existing bias, so they need to know where tufts evolved
Yeah.
in order to see if,
Pre-existing bias was
Exactly.
in effect. Yeah.
If tufts are ancestral, if they're found in all of these spiders, then it wouldn't be surprising to find all of these spiders with a preference. But if tufts are what they call a derived character, they evolve later in evolutionary time, there will be some members of the genus that have the tufts and others that don't. And those ones that don't should be expected to not have a bias towards tufts because it hadn't evolved yet. And And so they need to know where these spiders are in terms of their phylogenetic position, in terms of when they evolved, to determine whether tufts came first or tufts came later. And then they can look and see if tufts came later, they can look at ancestral species and say, okay, these things didn't have tufts, so if they do prefer tufted individuals, then they had a pre-existing bias for it. So they needed the phylogeny for two reasons. One, to determine the order of evolution, and then to identify which species they might test for pre-existing bias. And they, I don't know if were you gonna call attention to the caveat on page 171, right before choice of characters?
Oh, I don't think I have that.
Because the sample does not include
Yeah,
all of
I've
the
got
species in the
highlighted.
clade or the genus itself, this should not be considered the definitive phylogeny, but rather a preliminary analysis of the evolutionary relationships between these species. So they're already starting to issue caveats even back in the methods, like, this is a phylogeny that we made. It may not be the best phylogeny, but this is what we got.
And we see that kind of come into effect later, especially when you compare it to the Stratton paper.
Yeah, and so they also issue some caveats over in the results and some caveats in the discussion, and then we can talk about what the implications of Stratton are for this later.
So to begin the results, they talk about the habituation and priming behavior, and they say that females did not appear to show habituation or priming in their response to video stimuli. But how did they know that they weren't becoming primed to the videos? Like, what did they say how they knew that?
There was like no significant difference in receptivity.
From day to day.
And what would you expect if they were?
Like as time progressed, they'd be less receptive. Being observed.
So we look at figure 1, and what is figure 1 showing?
Just the overall receptiveness to each of the 5 different experiments. And both females, O. creata and ravneri. And the statistical differences between each individual test can be seen with the letters up top. Is that kind of
Yeah.
too
And then like
overviewish?
what level of receptivity is it showing? Like,
The proportion of females.
but like
I guess
are
it's like 100%.
Like from 0 to 0.8?
by saying female S. aureata displayed sexual receptivity most often to males with tufts and species-specific leg waving displays, and less often to males with species-typical displays but without tufts. So it's kind of saying that tufts, they're responding more to males with tufts even if they are showing the leg waving displays.
Like I have written to the side, like, a check, like they respond best to spiders with tufts and with leg waving,
Yeah.
and it's kind of moderately response to no tufts and leg waving, and then like the least amount of response to no tufts and no leg wave. In simple terms.
So that was, that section is the response for S. aureata. So then we have the response for Schizoposa ravneri, the females. And what is their data showing?
Preference for tufts, and then like an intermediate preference for the leg waving as well.
Yeah.
With no preference towards the control. Or the least amount of preference.
So it says that the response scores for S. ravneri males with tufts added were significantly higher than those for the S. ravneri control.
Right.
So they are also
They're
preferring
also-
tufts, which is what
Mhm.
it's showing.
Not only are they preferring tufts, what else is interesting about the response of ravneri?
Well,
They responded to leg waving as well.
yeah.
They responded to leg waving. They just responded.
To the, yeah.
They responded more to another species with tufts than they responded to control males of their own species, which I found very interesting. Because why would you prefer a male of another species than a male of your own species? that would argue strongly for what?
The grass is always greener.
That wasn't exactly what I was thinking.
Tufts play a significant role in female choosiness.
Yeah, and it would also indicate that chances are,
It wasn't pre-existing.
you know,
I mean,
it
The
was
pre-existing bias
was
Yeah.
pre-existing, and it would be a significant driver of
Yeah.
male tuftedness.
Mhm.
Now, we will call that into question later, but at this point in the reading,
Yeah.
in 1996, that would be strong evidence in support of McClintock and Hewitt's conclusions. In my mind, it does. The idea that females of one spider species actually prefer males of another species because they're tufted.
And that figure's saying that the S. ravneri that issue a higher level of response when the male stimulus includes a more obvious visual signal, whether that be the tufts or the heterospecific active leg webbing display. So then we look at figure 3, and what is it showing?
Latency of response.
Figure 3. Let me get to figure 3. Oh yeah. Okay.
Yep, so like Avery said, it's basically just showing that latency of receptivity response, it didn't vary significantly across the video stimuli.
But it looks like there is a variation, doesn't it? Because enhanced tufts in Ogriata seems low.
I was assuming, because it says that it didn't vary significantly, so I was assuming that meant that like the differences between the the S. aureata latency and the S. ravneri latency wasn't different for each
Female.
Like
Yeah.
for each like video.
not like overall.
Yeah, not like different among
So like
different
the
stimuli,
females,
but different
Like,
between.
like the
comparisons.
females of one species didn't significantly
Yeah.
vary another species within the control, within the no
Yeah,
tufts, within
that's
the
what I
enhanced
was taking it
tufts.
as.
Yeah, okay, I got
think
you.
I think what I found cross videos. Oh, cross videos do not. Okay, gotcha.
But it's also kind of brings to point the enhanced tufts being so low. I mean, they talk about this
Mhm.
early or later in the paper, but if say we haven't read later into the paper, and we
Mhm.
right now, they just mentioned that tufts had a big significant impact
Mhm.
on female choosiness, but so why wouldn't the enhanced tufts be higher?
Yeah, they
And that
basically-
would be
they
it.
But
basically-
higher latency would mean it takes
It
them
takes them
longer
longer
to
to
respond.
respond.
So
True.
enhanced tufts should
En-
be the lowest.
I guess-
Enhanced tufts should be the
They
lowest,
should be.
but actually they didn't find. They basically say at one point, just latency wasn't informative.
Mm-hmm.
And but when you look at the figure, like that looks like
It is.
it looks like it is
Yeah.
informative
Yeah.
because of course, what are they not telling you in these figures about the notations in the figures? And Elizabeth will know this because this is one of the things that I harp on in
they're
applied
not telling you
statistics.
Is it
What's,
which they don't have the letters up top?
No, they're not telling you what these are.
They're
Oh,
not
the
telling you what
confidence
the error
intervals?
bars
Yeah.
are.
Yeah.
Because my guess is those aren't confidence intervals. Those are actually probably either standard errors or standard deviations. And as a result, it makes it almost impossible to ascertain from the figure whether or not those things are actually different. What's that?
The captions are just kind of like,
They were
you didn't get
lacking.
anything from them.
Yeah,
It's
a
like
lot
one
of the
sentence per
descriptions,
each.
like what should be like figure captions, are kind of just like in the paper.
Yeah. So
True.
So that's yeah.
so oftentimes
Like, reference the figure, like, directly.
oftentimes when we have you guys, so let's just pause for a minute and talk about captions. When we train you guys to write figure captions, we basically tell you it needs a figure number, then it needs a title, and then it needs a description. And I think where you're finding these captions on these figures to be lacking is in the description, and it's because they've provided the description in the paper, and they don't repeat it in the figure caption. But it would be useful to have somewhat longer figure captions that did more explanation. Now they do give you letters on tops of the bars that basically allow you to see what's different from what, but having the accompanying kind of description that you see that they do a pretty good job of in the text of the paper and the results might actually be more informative. Somewhere along the line, I got trained that a figure and its caption should be able to stand alone. You shouldn't have to go and read the section on the paper to understand what the caption is doing. So my captions for figures that I write tend to have longer captions because they tend to be more explanatory. There's an exercise that I do in some of my classes where we go through and all we have is the figures from the paper. We don't read the paper. And give it to the students, and they work in teams to figure out what the what the figures are saying in combination with the captions. And we figure out what the paper is about, how the experiment was designed, and what they concluded from reading nothing but the figures and their accompanying captions. That's a good set of figures and a good set of captions if you can do that. These don't measure up in that way, and so you really do have to read the text of the results to get them. Continue.
So next they talk about the phylogenetic tests that they did and how they made the cladogram. So what did the phylogenetic tests tell them about the evolution of tufts? Kind of gave two options, and that's shown in the different A and B.
They say that the consensus tree suggests that tufts are derived character in the eukaryotic clade.
Mm-hmm. But then what are the two options they have for how tufts evolved?
They might have evolved independently twice, or just like it's in the ancestry common to Procriata and Chrysalis.
And then we're secondarily lost in S. floridana. So this is kind of addressed in the Stratton paper, as they have like a different phylogeny for the S. aureata group and how tufts evolved in that. Did you guys look at that paper?
Not really.
Avery avoided it because it had pictures of spiders in it.
Can you pull up the phylogeny that they made in the
Oh
Stratton
yeah,
paper?
sure. I can do that. My apologies to Avery. So, there are a couple of. This is going in exactly the opposite direction of how it works in my office, on my screen in my office. So let's make this full screen. And actually, let me just move this out of the way so we can all see well. So this is the phylogeny that is presented in the project. Do you want to talk us through this?
Yeah, so basically this is showing the two options for how tufts evolved. A is showing that it evolved twice separately, once for S. aureata and another time for S. crassipes. And then B is showing that and then was secondarily lost in S. floridana.
Why are somebody other than Elizabeth, why are these equally good explanations for how tuftedness might have evolved? You guys have you guys been exposed to how phylogenetic systematics works in other classes? Elizabeth is shaking her head. Yes. Did you guys go through this in
Planet Bio?
Yeah.
Bio? Avery?
Yeah.
Okay. So how do we assess explanations in phylogenies in terms of what one explanation might be better than another, or are they similar in their explanatory power? Okay, well, I know Elizabeth can because we've talked about this, so explain that to us.
Alright, so basically they both have two steps for how it got here. For A, there was one step where it was evolved for S. aureata and one step where it evolved for S. crassipes.
So let me pause there. So presumably back here, there was no tufts.
Yeah.
Yeah.
And at some point, tufts had to evolve. So tufts could have evolved here and here, which would give these two species tufts and not this one, because this one just continues to inherit non-tuftedness from its earliest ancestors. What's going on here?
Is that tufts evolved back there at that common ancestor, and then
These guys
these
inherited it from this ancestor.
and there, as Floridana, lost the character.
Yeah, so it was reversed. So in this case, there's one evolutionary transition for the development in Ocreata, a second evolutionary transition for development in Crassipes, and then no evolutionary change. That's just inheriting it from the ancestor. Here, there's a shift from, there's an evolutionary change from non-tufted to tufted, which is one evolutionary transition, and then another shift from tufted to untufted. That's two evolutionary transitions. So both of these scenarios have two evolutionary transitions, and so phylogenetic systematists would say those two explanations are equally likely. And they explain in the paper why one might be more likely than the other. I don't know that I necessarily buy that, but generally it's easier to lose a trait than it is to derive the same trait twice independently, is one thought. So they kind of favor this one. But that's not really what matters here when we look at these phylogenies. What's really important about this phylogeny from the standpoint of their hypothesis?
That the Rovneri didn't have tufts.
Rovneri didn't have
Uh-huh.
tufts, and it was derived, Rovneri evolved when in time?
Before tufts.
Before tufts appeared. Which then makes the argument for them
Pre-existing bias.
Pre-existing bias.
of this pre-existing bias. Yep, exactly. So when we go to their paper and they say this thing right here, did you have this highlighted? This is over on page
Yeah.
178.
I don't have like, yeah, I've got good portions of that.
Yeah,
underlined.
take it away, Elizabeth.
So they say perhaps the most striking finding of the research is that a high proportion of female S. ravneri show receptivity to conspecific males with tufts added and to heterospecific males with or without tufts. So
Even
they,
though these tufts are lacking in
Yeah.
conspecific males.
so then they're asking, like, could this be evidence of a pre-existing bias for a visual character? And that this pre-existing bias model suggests that male courtship displays and secondary sexual characters may evolve to maximally stimulate pre-existing female sensory and or cognitive systems. So I think what they're saying there is like the tufts evolved like they're agreeing with the pre-existing bias theories that tufts evolved to stimulate the female because they already had this bias for them.
Well, they didn't evolve to stimulate it, but they would not have evolved if the
With-
pre-existing bias hadn't been there. The pre-existing bias kind of sets the stage for a tuft when it appears to be preferred by females, and then that just sends you off on this pathway that we've already talked about when we were talking about sexual selection. Females prefer a particular trait. Traits that the males that have that trait tend to find more females to mate with, and then that reinforces the preference in the female, and it elaborates the trait in the male, and you're off on this runaway selection scenario of Fisher's from the 1930s. But then what do?
Then they give another little
Then they're like,
caveat.
Yeah, like,
we're
but
saying
wait.
this, but
But wait.
wait, we might not be right.
Mhm.
So
Because of their tree.
this,
it's-
yeah, their tree doesn't
Mhm.
have all the species of Ocreata in it, and so this conclusion is only as good as their tree is,
Mhm.
right? And they're very clear about that. Like,
Mhm.
this is a preliminary tree, it's not a complete tree, so this interpretation applies if this is the right tree.
Mhm. So we can see in Stratton that it's not.
So Stratton, which is one of George's students, well, this is from another day, who's one of George's students, goes and builds a better tree based on way more characters and way more species within the genus. And this shows the Ocreata clade here, and this shows where dark pigment on the legs occurs, and this shows where tufts occur. And we don't really need to worry about the entire tree. What we need to worry about is the the Ogriata clade, which is what this is.
So the important part of this is where S. ravneri occurs in the tree.
Here, here, and there. Hmm?
So you can see.
Tuftedness evolved way back here,
Mhm.
which is why Ogriata and Craspes are tufted, which is why Stridulans is tufted. But ravneri is a relatively recently evolved, according to this tree once again. And all phylogenetic trees are hypotheses; they can be disproven by later data, as we're now seeing. It appears from this that the presence of tufts is something that is relatively common within the Ogriata clade, and ravneri, Udzai, Floridana, those are all kind of exceptions to the general rule. So would ravneri be a good species to look at to test the pre-existing bias hypothesis? I'm sure this was a question that Elizabeth was about to ask you, but I've
No,
hijacked
because they're
her.
already evolved, right?
No, because tuftedness had already evolved
Yeah.
at that point. So in their phylogeny, they found it to be one of the ancestral groups that evolved before tuftedness had evolved. In that case, a preference here would be seen as evidence for pre-existing bias. But if you have a better, more complete phylogeny, you find that ravneri is embedded within this clade of tufted individuals. Well, then it would make total sense for ravneri to have a bias towards that,
Yeah.
because it evolved from tufted ancestors.
So what should they have used if they'd had this complete morphology? Which ones?
Duplex or something.
Duplex or
That one's down there.
whichever of the.
Duplex or
those.
saltatrix or crassipulpata or humulus or avida or macucae or communis or maxima or cespitum or retursa or chiricahua or mimula
Salsa.
or minnesotensis. Any of those
Salsa.
you could have. And there's not a salsa.
Yes,
there
there's
is,
too.
but it
It
has
has the
tufts.
tufts.
It has tufts. You
I
wouldn't
just
want to
thought it
use
was
tuft.
funny. I'm sorry. I also couldn't tell if it was blacked out or not.
So you could have used any of those, but what you shouldn't be doing is you shouldn't be using ravneri, Udzai, or Floridana, because they're embedded within a clade of tufted species. No surprise that they might have a bias toward tufted males, because they evolved from ancestors that had tufted males. This was published in 2006. McClintock and Utes had no idea what the true phylogeny was when they published this paper.
Has this
And-
changed at all? The
Pardon?
Has this changed at all? The tree?
Oh, I think this is actually still to date the most complete phylogeny that exists for this genus. And this phylogeny also doesn't contain all of the species of Schizocosa. So somebody could do a more complete survey of Schizocosa, base it on either morphological characters or molecular characters, and the topology of this tree might change again, in which case we could go back and reevaluate McClintock and Utes. Was this a molecular phylogeny or a morphological phylogeny? I think maybe Elizabeth's the only one that looked at it in any detail.
I'm also not sure which Stratton did.
It's based completely on morphology.
Okay,
What's
that's
the-
what I was thinking.
What's the trick with using morphology in this genus? We've talked about it in the time that we've been
Because they
sitting
look
here.
extremely similar.
They look all very similar to one another, so it's
Hard to
very
decipher, yeah.
it's very difficult to decipher who's related to who, because it's a genus that is only found in North America. It's presumably not been around for tons of geological time, so it's diversified very rapidly. That's one of the reasons that everybody looks similar, and that's one of the reasons that there's so much reproductive compatibility. If you can get them to mate, they can produce hybrid offspring among the different species. And so all of that points to a relatively recently evolved group of spiders. And that makes doing phylogenetics really difficult because of the morphological similarities, the potential for gene flow from one species to another, etc. So what would one thing you might want to do rather than making a morphological, using a morphological data set to build a phylogeny, what might you want to use instead?
Use a molecular data set.
Yeah, you might want to use a molecular data set where you look at differences in DNA sequences between species, because those things are going to have way more markers that vary from species to species.
And no one's done that yet for schizocosa.
No, because it's like 38 species and it's going to take a ton of money, and who really gives a
Yeah.
crap about a bunch of spiders having sex in the forest?
That's a good point.
That's a funny way to put it.
But it would be a fascinating thing, because of this uniqueness of all of the behavioral mechanisms that are important in keeping these species apart. What else did you have for us?
I had some notes on female choice.
Oh yeah,
Um,
hit us with it.
So they talk about because the natural variation in tuft size may influence female choice, and they couldn't really test for the influence of these factors because you have to show the females video images of several males that display at different rates. So how do they? That's kind of how they suggest for testing for female choice. But how else do they? They talk about testing for female choice in regards to tuft size.
What page are you on? Oh, there it is right there.
178.
Gotcha.
They kind of give a direct suggestion of how to test for female choice.
So like present them with, like their conspecific males without the tufts and like whipping.
Heterospecific male with both tufts and like waving.
So they kind of want to see if like,
Just kind of flip the script.
yeah,
Yeah. Flip the attributes with the males.
yeah. And they also want to say, they say one needs to show females video images of several males that display at different rates with digitally reduced or enhanced tests and measure their response.
Because they said they use like the same male
Mhm.
for all the videos.
They
Yeah.
did.
But
Because they didn't-
just controlled his characteristics.
Yeah, so they couldn't see how
But
the leg
behavior was
waving
the same, yeah.
affected female choice.
Yeah, so, so what they're trying to get at here is that it may be that those males that have larger tufts are also just better in quality in a lot of ways.
Mhm.
As a result, they display more vigorously, more often, et cetera, et cetera. That's not something that they've studied in this case because they used the same male and they just manipulated the tuftedness of the male. But there might also be other things other than just tuftedness that might be correlated with tuftedness that are just as important. So if you have tufts and you're just sitting there, so I'm now imagining spiders at a bar, and so you're at a bar and you're a tufted male but you're shy, and so you keep your front legs down and you're just sipping your beer and everything else, and 3 bar stools down there's a male and he's like, hey, look at me, obviously one of them is going to have better, well, okay, maybe not have better success than the other, simply because one is displaying actively and frequently whereas the other one is not. And so that doesn't factor into any of this. We see in birds oftentimes, so going back to the bowerbirds, when females go by and look at bowerbirds, they've shown that the females are looking for specific things. They're looking at the quality of the bower, they're looking at how much stuff it's adorned with, and they're also judging the vigorousness and the stamina of the male as he dances around his bower. All those things point to male quality. If you have a vigorous dance, that's a good sign that you're a vigorous male, especially if you built this huge ass bower and spent a ton of your time and energy going and gathering all this crap from the forest and hanging it on your bower. Those are all correlated with, presumably in the mind of a female, high genetic quality, and so I'm gonna mate with you. This other guy over here, ratty looking bower, not many sticks in it, not much pieces of blue plastic on it, and you're just kind of lackadaisical about your dance, I'm not mating with you. You're a bum. So those things are important, and so this study doesn't address that. You would have to make a new set of stimuli. It did vary not only in the toughness but also in the frequency of display, the magnitude of display, et cetera, whatever they thought was important to get at these other non-tuft related aspects of the display.
I imagine when reading through the kind of methods of how they went about doing the video tests that you could probably do it with extremely more efficiency than in 2022 than in 1996.
Oh yeah.
Like they were using like their camera
Yeah.
descriptions. I was
Yeah.
like, what is this?
Yeah.
Oh yeah. So
And it might make
Yeah.
it more realistic. That could also could potentially like change results. or
yeah,
make it
and
easier.
when you look, I was looking for actually videos of the camera, of the wall. Do you guys know do you guys even know
A
what
Walkman?
a Watchman is?
I know what a Walkman is.
Okay, so a Walkman
Yeah.
is a stereo.
Yeah.
I used to have one. It was waterproof. It was yellow. I was slick. It played cassette tapes. A Watchman is the TV equivalent. So it had like a little 2-inch by 2-inch screen, had an antenna, and you could get broadcast TV, just like if you were in your house, but you were out jogging. I don't know why you would be jogging and watching TV at the same time, but whatever. It was TV on the go, and they were kind of rectangular, and what they basically did was they created this arena, and they just kind of taped a Watchman up to the thing, so that only the screen was poking up above the thing, and they would just play their videos back. They hooked it up to a video cable and played their videos back on this little 2-by-2-inch screen. So yeah, we have way better ways of doing this nowadays than they had back then. But at the time, this was like cutting edge stuff.
Yeah.
Because by just removing or adding to the single video that they have of one male, you remove all variation among individuals from the equation. The only thing that is different about those videos is
The size or not.
tough presence and size, which is pretty cool.
Yeah.
And George had a postdoc who carried on with this, doing these types of studies with jumping spiders, and you can see. I noticed this in seeing these presentations at meetings and stuff. The quality of the video manipulation got better and better and better until by the time you're into the mid 2000s, seeing these presentations at meetings, it's almost like, almost Disney-like quality animations of spiders doing their thing, and it does get better, but at the time, this was cutting edge. Cutting edge behavioral science, not so cutting edge phylogenetic systematics.
Yeah.
And that's fine, but it points to the need for knowing. We talked early in the semester about the comparative method. This is one application of the comparative method, and in order to do the comparative method, to employ the comparative method adequately, you have to have a good phylogeny to rely on. Your comparative analysis is only as good as the phylogeny that you're working off of. And so sometimes, departments, as scientists get older, they're systematic biologists, the people who build phylogenies for organisms. Those people will retire, and departments don't replace those people with systematists. They replace them with something else, a behaviorist, for instance. But the problem with that is that then that removes this important part of science that we don't probably pay enough attention to here at Williamsville College, which is that these evolutionary trees, these images that we show, that show how organisms came into being over evolutionary time, they actually do have utility for people other than systematists. And in this case, Gail Stratton, she is not a systematist. She is a behaviorist. She came up in George's lab also. But she felt that there was a need for having a better phylogeny for this group. No systematists were doing it, so she was just like, all right, then I'll do it myself. And so systematic biology is in some ways a subdiscipline of biology that is kind of in crisis, because a lot of other subdisciplines don't really think that it is important, but it actually is crucially important because a lot of other fields can't do anything without that information. Or they can do things, but they can't do as interesting things without that information. And this paper is just really good at pointing that out. else do you have to add? What was it?
The last comment I had was they gave another caveat in their conclusion
Oh, yeah,
where they
where's that?
say, it's like the very last line, they say that it's possible that female preference for tufts may have evolved prior to the tufts themselves. Further research will be required before a definitive mechanism for evolution of decorative leg tufts and leg waving displays can be accepted. So basically, like, here's what we think, but also, maybe not.
Yeah, they did that a lot.
Also, Gail Stratton, please go build this file out.
Yeah.
I mean, they didn't say that to her
But-
specifically. And I haven't talked to Gail in a while now. I'm not sure exactly what precipitated that, but it might have just been that she just finally got tired of not having a phylogeny for the group of spiders that she works with. And she's like, well, nobody else is going to do it. I'm going to do it. And then she just did it. But yeah. Molecular phylogeny would be really interesting for this group because of all the reasons that I talked about earlier. But I think there are now 38 species described in the genus, and they're scattered all over the United States. But one of the cool things is that there are western species, and there are eastern and southern species, and then there are like species in Canada, and there's this chunk in kind of the montane west where there aren't any members of the species, and so having a good phylogeny that also is geographically complete would be interesting because you might see this divergence between eastern species, western species, and northern species that are kind of separated by this dry area in the kind of western United States. So, anyway, and we're finished a couple of minutes early, and that's that. Good job. Good job on your guys' part in terms of being prepared.
Good job, you did good.
And I will take your summaries. Where's your summary?
Is this
It-
one
Oh, there it
inch?
is. Okay, cool.
Mhm.
What?
I swear, because I remember you noted in my last summary that it wasn't 1 inch, and then I made double check sure,
No, that's fine.
but then now I'm looking
Yeah.
at Elizabeth's and it is not
Well,
the same.
mine's in this is in the header.
Right.
Yeah.
And then,
I don't know, I set my margins.
because
I'm less concerned with the 1 inch than I was about the font, because
Oh,
actually
yeah.
what I just gave you was I gave you all the formatting instructions.
yeah.
So, yeah.
I just realized this is stapled backwards, so this is the front page. Okay.
I'll figure it out.
Okay.
Yeah.
I went to read the first line and I was like, that is not correct.
The reason I do this is I want all of you to be writing for the same length goal, and I can't assess that
Yeah.
if they're not similar.
Yeah, no, that's
at
fair.
least. I'm not a big stickler about format, but when they're radically different, I can't assess that particular aspect of it.
Yeah, that's fair.
Yeah. I didn't take anything off of it because of that, but it just helps me assess.